That's not right. Hi, before we begin, are there any questions? Here we are. At least I hope that's where we are. All right, I'm not hearing anything. Can somebody confirm you can hear me?
We can hear you.
Great. Sorry for being slow or late today. My computer is acting up and I'm having trouble with it. Well, let me share my screen. Today is February 27th, and we are supposed to be finishing chapter 8 and starting chapter 14. We're behind schedule, and I don't know if we're going to finish chapter 8 or not today. Hopefully, we'll at least get close. Any question about any of that? You should also look at the grading. I'm not going to talk about it now, but I did get several assignments graded in the last couple of days, including Lab 7. And I think one or two lecture assignments. There is a quiz tomorrow, quiz 4, covering Lab 7 and chapters 5 and 6. Any questions about any of that? All right. Let's get out of here. I should probably shut that down. On my notes, I've got that we had discussed the regulation of bacterial genes. And this is the last slide we had talked about. So cells want to regulate their contents, and one way to regulate their contents is to regulate the control of gene expression, meaning turn the gene off if you're not going to need it and turn it on if you are going to need it. We're only going to be talking about inducible operons. An operon is a model of gene expression. Bacterial operons do contain the gene of interest. Let me see if I can blow this up. So when we're talking about the lactase gene, the gene that codes for the enzyme lactase, which metabolizes sugar lactose, that would be in the lac operon, and it would be the Z gene. That is a structural gene, a gene that directly codes for a gene end product, in this case, the enzyme lactase. Also in the prokaryotic or bacterial operons, we have controlling regions around the gene of interest. And these regions control the expression of the lactase gene or the structural gene of interest. And that would be the promoter for one controlling region and the operator, another controlling region. And then something that isn't shown here, but at the end of the gene, there would be the terminator. All of these are controlling regions that help control the expression of the gene of interest, the Z gene, which is the lactase gene. In the bacterial operon, we also have other structural genes. They do not It depends on which operon we're talking about, but in bacteria, you may have more than one structural gene in the operon. In the case, there is more than one structural gene in the operon. They are all related genes. In the case of the lac operon, the Y gene codes for the transport protein. Come on, mouse, stop dying. The Y gene codes for the transport protein that brings the sugar lactose into the cell across the cell membrane. Like I said, there's something wrong with my computer. I'm having a real difficult time getting this computer to work. So the Y gene is involved in the metabolism of the sugar lactose. Doesn't directly metabolize it, but it brings the sugar into the cell. The A gene is the gene that helps convert the sugar beta galactose to the sugar glucose. And that too is involved in the metabolism of the sugar lactose. If you use the enzyme lactase on the sugar lactose, you get the sugar glucose, which can be used by any cell because it's the base sugar, and the sugar beta galactose. Beta galactose has to be converted into the sugar glucose before the cell can use it. And the A gene is a gene that helps convert that sugar, beta galactose, to glucose. So all of these genes are involved in lactose metabolism, and they're all related to each other, and they're all in the lac operon, which is the operon is sort of the super gene controlling the lactase gene. Any question about any of that? The lactose operon is an inducible operon, meaning it's normally shut off, but when the cell needs it, it will be turned on. And that's what inducible means. We'll talk a little bit more about that in a minute, but I do want to briefly mention about eukaryotic operons. They are similar to bacterial operons, except a eukaryote operon only has one structural gene in it. Eukaryotic operons also have more regulation of the gene expression. So they'll have more regulators controlling the expression of this Z gene. Come on, mouse, stop dying. So obviously humans have the sugar that not the sugar, the enzyme that codes for lactase. So we do have a lactase operon, but we also have the transport protein that brings the sugar lactose into the cell, but it is on its own operon. It's not with the lactase gene. And then we have the same gene that helps convert the sugar beta galactose to the sugar glucose, but it too is in its own operon. Any questions about that? We're only going to be talking about bacterial operons, and to tell you the truth, we're only going to really be talking about the lac operon. The operon in bacteria, particularly E. coli, that contains the lactase gene. All right, any questions about that? Let me state, oops, I lost the, let me state that there is a regulatory gene that does control the expression of the lac operon. The regulatory gene is not a part of the operon, but it does regulate the expression of the lactase gene or the lac operon. It is its own operon, meaning it will have a promoter upstream of the regulatory gene, and it'll have a terminator at the end of the regulatory gene. Normally, the regulatory gene is nowhere near the gene that it regulates. In fact, on eukaryotes, normally the regulatory gene would not even be on the same chromosome as the gene that it regulates. In the case of E. coli and the lac operon, though, the regulatory gene is right next door to the gene it regulates. And we'll talk about how the regulatory gene regulates the lac operon in just a minute. And like I said, remember the regulatory gene is its own operon, and it is a different operon than the lac operon. The lac operon is an inducible operon, which mainly means that it is shut down. The E. coli cell does not usually have the lac operon turned on so that the lactase gene is not made. Can anyone tell me why E. coli might want to normally have the lactase gene or that the lac operon shut down? Anyone have a guess? Well, what do you know about the sugar lactose? Well, think about that and we'll move on. The gene is normally turned off, and how it's turned off is the regulatory gene is expressed, and that's this I right here, and it makes a messenger RNA, which then is translated into a protein. This protein is a regulatory protein, and it is a repressor protein that will bind to the operator of the lac operon. When this repressor protein is bound to the operator, then when the RNA polymerase binds to the promoter and moves down the DNA, the RNA polymerase will be blocked by the repressor protein. If it's blocked, it cannot transcribe the Z gene, and then the Z gene will not be transcribed, will not be translated. We will not have the enzyme lactase in the cell. And this is normally how E. coli shuts down the lac operon. Does anyone have a guess why E. coli normally has the lac operon shut down? I'm looking to see how many people are online. Come on. I can't seem to get my computer to show it.
Does it have anything to do with like the way that we use energy? Like normally we use glucose and we don't want to be using lactose all the time.
Well, that would be for us. We're talking about E. coli.
Oh, right.
But you
Right,
are
right.
correct in the sense that glucose is the preferred sugar. So if ever glucose is around, E. coli would prefer to use glucose and not galactose. I mean, not galactose, not lactose. So that is true. But what else do you know about the sugar lactose? Like, where do we find the sugar lactose? All right, think about it.
Milk?
Yeah, we find the sugar lactose in dairy products. How often is E. coli going to be around dairy products? Talk about E. coli in the ground or on an apple tree. All right, think about it. We'll move on. So the lac operon is normally shut down because the repressor protein binds to the operator, preventing the RNA polymerase from moving down the DNA and transcribing the lactase gene, the Z gene. However, we have to have a way of turning on the lack operon, because if ever the E. coli is around the sugar lactose, it'll want to metabolize the sugar lactose. This would be especially true if there was no other sugars around. And that would be something like an E. coli on a dairy farm, and it happens to fall into a bucket of milk. The only sugar it's going to find is the sugar lactose. E. coli would then want to turn on the lac operon. How it does that is it has an inducer which activates the lac operon. Whenever the sugar lactose is around, we have the sugar allolactose around. Allolactose is an isomer of lactose. That means one is a left-handed molecule, the other is the same molecule, but it is right-handed. Hopefully you guys remember from your preliminary course what an isomer is. The point is, whenever the sugar lactose is around, allolactose will be around. Allolactose is the inducer. It will bind to the repressor protein and inactivate it. If the repressor protein is inactivated, it will not bind to the operator. The little video I'm going to have you is a little bit mistaken on this, and that is the repressor protein the repressor protein and the inducer, allolactose, only bind to each other when they both are free. When the repressor protein is bound to the operator, when the repressor protein is bound to the operator, the inducer, allolactose, cannot bind to it. And the video is a little bit off on that. It had the repressor protein on the operator and the inducer binding to it, inactivating the repressor protein. It doesn't really work that way. What happens is that the repressor protein will bind to the operator, but that binding is not permanent. It's a stoichiomic reaction, and every once in a while, the repressor protein will fall off. If there's lots of allolactose around, it will bind to that repressor protein that falls off. And then the free repressor protein is bound to the allolactose, so there will be no repressor protein now that can re-bind to the operator. And that's how the inducer allolactose turns on the lac operon. It will prevent the repressor protein from binding to the operator. And if it's already bound to the operator, sooner or later it'll fall off, and then it'll bind to the inducer, and there will be no active repressor protein capable of binding to the operator. And then when the RNA polymerase binds to the promoter, it'll move down and transcribe the Z gene, the lactase enzyme. And that is how we turn on the lac operon. And it is turned on, and that's why we call it an induced operon. Whenever the sugar lactose is around, allolactose will be around, and that will turn on the lac operon. Any question about any of that? If there's no questions, let's watch the little video on the lac operon.
The E. coli lac operon is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into sugars used for cellular metabolism. This inducible system also involves bacterial DNA, a repressor, mRNA, and the sugar molecule lactose. This animation will only focus on 2 of the 3 proteins encoded by the lac operon, beta-galactosidase and permease. Gene expression can be induced or turned on when a specific inducer molecule appears in a cell. For inducible systems, a repressor molecule prevents gene expression by binding to the upstream controlling region.
That's the operator.
Lactose is the lac operon inducer molecule. After first appearing in the cellular environment, lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from the controlling region. At this point, RNA polymerase can begin transcription of the operon. Here we show 2 of the 3 lac operon genes being transcribed into mRNA. Ribosomes then bind to the mRNA, and the 2 proteins are translated. The first protein is beta-galactosidase, which breaks down lactose into 2 simple sugars. The
That's
second
lactase.
protein is permease, a membrane-bound protein.
That's
When
the
embedded in the cell membrane, permease functions to provide a direct route for the lactose outside the cell to be imported into the cell. This import occurs at a much greater rate than the passive transfer we first observed.
Okay, permease is the transport protein that brings the sugar lactose into the cell. And it gave a different name for the sugar or the enzyme lactase. I think it called it beta-galactosidase or something like that. It's the same enzyme as lactase.
Because translation continues inside the cell, other permease proteins become embedded in the membrane. This further increases the rate at which lactose enters the cell. Beta-galactosidase breaks the cellular lactose into the simple sugars glucose and galactose. Once its concentration is greatly reduced, the lactose bound to the repressor are released. At this point, the repressor again binds to the controlling region and gene expression is halted. For all inducible systems like the lac operon, it is the interaction of the repressor and inducer molecules that mediate gene expression.
All right, any questions about that video? So when you make the messenger RNA, when it's transcribed, what do you notice that's different about this messenger RNA? We've never seen this before. Anybody? It's pretty obvious. This messenger RNA encodes for 3 different proteins. We've never seen that before. That's the lactase gene, also called beta-galactosidase, the permease gene, which is the transport protein that brings the sugar lactose into the cell, across the cell membrane, and then transacetylase is a gene involved in converting the sugar beta-galactose to the sugar glucose. And when that messenger RNA is made, all 3 of these proteins will be translated. And that's what happens in the lac operon. Now, a bacterial operon may have 1 structural gene or several structural genes in the operon. If there are more than 1, they are all related to each other. In the case of the lac operon, all of the genes are related to the metabolism of the sugar lactose. Any questions about any of that? Can anyone figure out why E. coli normally shuts down the lac operon? Well, think about that. All right, let's move on. We're going to talk about mutations. A mutation is a change in the genetic material, such as a change in the sequence of the DNA. Mutations may be neutral, beneficial, or harmful. And I will point out that not very many mutations, meaning that it's fairly rare when a mutation is beneficial. I do remember reading about one, and somebody had a fruit fly lab, and they had in jars, it wouldn't be a jar like this, but close enough to it, a jar where they kept the fruit flies in it, and at the bottom of the jar there'd be media where the larva would eat, and then the fruit flies would crawl up, the larva would crawl up on the edge of the jar and then make a pupa. And after the pupation stage, the pupa would kind of hatch, and then the fruit fly would be born and would fly out. Well, the person who was running the lab was very lazy, and he would just not change the jars. And so the whole side of the jar would be covered with pupas. And eventually, the fruit flies would not be able to find a place to crawl up as the larvae and find a place to pupate, because the guy just never changed the jars. And so he would continue getting flies and continue getting larvae, but obviously the pupa would stop, and so there'd be no new fruit flies. And what happened was, this jar, you have a cotton swab and you put it at the top, sort of like that, and that's how you close the jar. And there was a mutation in the population, and the larvae were able to climb up the jar and then climb onto the cotton swab and then pupate on the cotton swab. And that mutation was really selected for in this lab because the person never changed the labs, and so there was no place on the side of the jar for the pupa to be and go through the pupa stage. And so that was a beneficial mutation. It did increase in frequency in this person's lab because he was lazy and he just never changed the jars. Normally, mutations are either neutral or harmful. And it's usual thinking that mutations are harmful, but there are a lot of mutations which are neutral, and you'll see why in just a minute. A mutagen is an agent that causes a mutation. However, mutations do not require a mutagen. They can happen spontaneously, and we call these spontaneous mutations. Spontaneous mutations occur in the absence of a mutagen. Any questions about any of that? All right. There are different types of mutations. Probably the most frequent mutation is a base substitution, where one base is changed, and this is also called a point mutation. We have three types of base substitutions. There's a silent mutation, a missense mutation, and a nonsense mutation. All right, there's also a frame shift mutation, and that is different than a base substitution. A frame shift mutation adds or subtracts a nucleotide from the DNA. And you may add or subtract more than one nucleotide. If you subtract well, let's say you add a nucleotide to the DNA sequence, there will be one more base in the DNA. And more importantly, where you had a codon of 3 bases, you will have the same codon, but obviously one of the bases will switch. And so that will change that amino acid, or it may change that amino acid. Everyone see that? But what's more, when you add a base, it changes the reading frame of the codon because the codons read by 3. Is that clear to everyone? So like if you have 27 codons, that means, what is that? Something like 9 genes, if I did my math right? 9, excuse me, 9, not genes, 9 codons, 27 nucleotides, something like 9 codons coding for 9 amino acids. Let's say at the very first codon, we add an extra amino acid. This codon now is not 4 nucleotides, it'll be 3. And it could change the amino acid because let's say we had, well, the first one's going to be AUG, and it's switching to, well, we can't change AUG because it's got to start the protein. So let's make the 4th codon different. We add it at the 4th place. So we have AUG, and let's say the next codon is also AUG, but we now add a new nucleotide, and let's say we add A. So that codon, instead of being AUG, will be AU, because we read the codons by 3 nucleotides. And then the G will start out the next codon. The point is, when you add or subtract a base, at that point, you change the reading frame, and all other codons downstream of that may be changed. If it's a different nucleotide sequence, it will change the codon. Any question about that? So a frame shift mutation, wherever it happens, downstream of that mutation, all of the codons will be different. And that's a real change in a protein. A macromutation is a subject that is not discussed in your book. So you'll have to pay attention to this lesson for macromutation. A macromutation is where an entire region of a chromosome is either added or deleted. And you can find 2 different definitions of a macromutation online. There's the one I gave where there's a massive change in the DNA, where a whole region of a chromosome or an entire chromosome is either added or deleted, a macromutation. But in the online, you can also find that macromutation means a massive change in the expression of genes, usually involving a controlling gene that controls other genes. And we're not going to talk about that type of a macromutation. We're talking about the change in a large number of nucleotides. An example of a macromutation is Down syndrome patients, which have an entire extra chromosome. If I'm not mistaken, they have 3 copies of chromosome number 23. Another macromutation would be an individual born, like a female individual born with either only 1 X chromosome or maybe with 3 X chromosomes. And then there's the hermaphrodites, which are born with the chromosomes X, X, Y. All of these are macromutations. Usually, macromutations are lethal in at least the human species, meaning if someone is born with 3 chromosome number 1s or only 1 chromosome number 1, that will be lethal. And usually that'll be lethal in the embryo or fetal stage, meaning the baby will not be born. It's so lethal. But there are some like an extra X chromosome or an X chromosome missing, or XXY individuals, or obviously Down syndrome, these individuals can be born, and these mutations, the macromutations, are not lethal. The reason why XXY is not lethal is because XX codes for a female, and then XY codes for a male. And so XXY, I guess, codes for something in between, meaning a hermaphrodite, and that's not lethal. X individuals can be born. They only have one X. They're usually developmentally delayed, slightly, what's the word? Mentally slowed. There's another name for that, but I forgot the name of that. Meaning usually they have difficulty with the functioning of their brain. They're not usually as bad as a Down syndrome patient, which they are mentally retarded. And there's another name for that. I've forgotten it. The mentally retarded is the older name. That's a little insulting. But the reason why the Down syndrome patient can survive is with 3 chromosome 23s, is this is one of the smallest chromosomes in the human genome. And apparently having 3 is not lethal, although it does affect the development of the individual. Here was a case I read about, and this boy, I guess we have to blow that up so you can see it, was having spoons sticking to his chest, and they were wondering, was he a mutant and possibly becoming a Magneto, if you know about that. I never heard further, but they were testing to see if he was a mutant, and that's why the spoons were sticking to him. Some people wondered if he was just born with sticky skin. Other people were wondering if he had a magnetic ability and the spoons were being attracted to him. I never got an update on the article about what the case was and what the studies were, but they were studying him to find out why spoons were sticking to him. And he's a possible mutant. We don't know yet. And like I said, I never found an update on the article. All right, let's talk about the different types of mutations. Base substitution is where one base is substituted for another base. And in spontaneous mutation, when DNA is replicated, the DNA polymerase can make a mistake, where instead of putting the correct nucleotide on the G, it added a T by mistake, and that mistake happens not very often, but something like 1 in 10 to the 9th bases that are replicated. So what is that, 1 in a billion bases? And before this DNA replicates, the cell can correct that mistake. It would assume that the mistake, because this is not pairing correctly. Come on, mouse. This is not pairing correctly. So it tells the cell there is a mistake there. And the cell will always pull out the T and then re-put in, meaning the polymerase will come in there and fill it in. And most of the time it would put in the correct C. But once this replicates, the mutation becomes fixed. Actually, this strain would be fine. It'll be replicating fine, but this T will not replicate with a G across from it. It'll replicate with an A. And now the mutation becomes fixed. Any question about that? This is a base substitution where one base is substituted for another. And in this case, it does result in a different amino acid being translated, meaning the different codon codes for a different amino acid. Any question about that? Now, with base substitutions, we can have 3 types of them. There's a silent mutation, where you change the DNA nucleotide base, but it does not result in an amino acid change in the protein. What happens is the nucleotide changes to another codon, which codes for the same amino acid, such as UUU, come on, mouse, changing to UUC. This is a change in the DNA, but it does not result in a change in the amino acid in the protein. And that's why we call it a silent mutation. The DNA is changed, but the protein is not changed. No effect on protein function. Any question about any of that? And this is the main reason why a mutation can be neutral, because the DNA is changed, but the protein is not changed. And that's a neutral mutation. There could be other changes which do code for a different amino acid, which could be neutral. One example is when we look at the blue-eyed allele of humans and the brown-eyed allele of humans, today, there is no difference in the reproductive rate of the blue-eyed allele versus the brown-eyed allele. And 10,000 years ago, in Northern Europe, there was a mutation in the human population, and it resulted in a base substitution, which was not neutral. It did change the protein, and it resulted in the blue-eyed gene, resulting in blue eyes. Now, 10,000 years ago, they think that the blue-eyed allele had a reproductive advantage. The reason why they think that is the blue-eyed allele started from essentially zero in the population of Northern Europe to it's now the majority. It's over 50% of the alleles in Northern Europe. And so they think it had a reproductive advantage 10,000 years ago, but today it does not have a reproductive advantage. What that advantage may have been, we do not know. It is possible the blue-eyed allele happened to be next to an allele that had a reproductive advantage and that carried the blue-eyed allele up in frequency, but it would be hard to explain why it became the main population in Northern Europe unless it had a reproductive advantage. Any question about any of that? All right, there's a missense mutation, and I've kind of already talked about it, because the brown-eyed allele, which was what all people in the world were until the blue-eyed allele mutation occurred 10,000 years ago in Northern Europe, was a base substitution that was a missense mutation. A missense mutation changes the DNA and changes the amino acid in the protein, resulting in a different protein. In which case the protein gave the individual blue eyes as opposed to brown eyes. So here we're looking at it here. Let me see where this is. That looks like the protein that was changed. So instead of being GGC, it's changed to AGC, coding for a different amino acid. And there we have a different protein, a missense mutation. Any question about any of that? So a missense mutation is just a base change that changes the DNA, but it also results in a different codon, coding for a different amino acid. And that's a missense mutation. Any questions about that? An example of a missense mutation is many sickle cell anemia patients have a missense mutation where one codon changes one amino acid in the protein, resulting in the sickle cell protein of hemoglobin, which is only one amino acid change out of thousands or more than 1,000 amino acids. And it totally changes the protein, the hemoglobin protein. It doesn't work as well as the normal hemoglobin protein. And the other example is the blue-eyed allele gene, causing the blue-eyed phenotype. And I don't want to talk about why it's blue-eyed, but it was the missense mutation from the brown-eyed allele. Question is, will humans ever have another mutation? Like dogs have blue eyes and brown eyes and yellow eyes. Is it possible that there will be another missense mutation in the human population resulting in yellow eyes? And I'm guessing it's a possibility because dogs have it. Any question about any of that? All right. The third base substitution is a nonsense mutation where one nucleotide is changed in the DNA and it changes the codon so that instead of coding for an amino acid, it codes for the stop codon. And that's why it's called a nonsense mutation. Instead of coding for an amino acid, you now code for the stop codon, and that will cause a truncated protein. So this is a very short protein, 1, 2, 3, 4, only having 4 amino acids. If we change the nucleotide here, we then change this codon. Instead of coding for the amino acid lysine, it now codes for the stop codon, and we have a truncated protein. Nonsense mutations are usually pretty severe, and that's because the protein is much smaller. It usually does not function or does not function as well if it does function at all. Any question about any of that? Some examples of a nonsense mutation is a mutation causing muscular dystrophy and cystic fibrosis patients. Some of them have a nonsense mutation where the DNA is changed, and that results in a change in the protein because the amino acid is not coded for. It's a stop codon, resulting in a truncated, smaller protein. Any question about any of these? All right, let's talk a little bit more about the frame shift mutation. This is where you insert or delete one or more bases. And maybe I can go through this in a little better example. Normally, we have AUG coding for methionine, this codon coding for lysine, this codon coding for phenylalanine, this codon coding for glycine, this codon coding for the stop codon. If we now, what have we done? Taken out an amino acid? Not an amino acid, a codon, not a codon, a nucleotide base. AAA, yeah, we have 3 AAA here. We remove 1A. Now reading the codon, we still have AA. And to get the third one, you borrow the nucleotide from what used to be the next codon. So this becomes AAC, coding for a different amino acid. Instead of phenylalanine, we code for leucine. However, the next codon is now not CCG because that first C is pulled up to the codon, the third codon. So the codon will be read as CGA, which codes for a different amino acid. And then there won't be a stop codon here. It will continue reading T, and whatever the next nucleotide is. And you will, in this case, continue reading the messenger RNA until you reach the stop codon, or you reach the end of the messenger RNA, in which case the ribosome will fall off the messenger RNA, and then protein translation will stop. Any question about any of that? There is one insertion or deletion which is not a frameshift mutation. And let me blow it up here. If we were to pull out all three of these A's, that would not be a frameshift mutation because you still have coding methionine, lysine. You now don't code for phenylalanine because the AAAs are pulled out. But then you have glycine and then the stop codon because the downstream codons are read correctly. You just get one codon pulled out. So whenever you insert a number of bases that's a multiple of 3, which could be 3 or 6 or 9 or whatever, or you pull out a multiple of 3, it is not a frameshift mutation. It is a mutation, but it is not a frameshift mutation because you do not change the reading frame of the codons. Any question about that? All right, some Tay-Sachs patients are that because they have a frameshift mutation. Any question about that? All right, for the macro mutation, maybe the best example of that is a Down syndrome patient, where the patient is born with 3 chromosomes 23. When we're talking about the causes of mutation, it's important to realize there are 2 causes. There can be spontaneous mutations, simply when the DNA polymerase makes a mistake, and there can be mutations caused by a mutagen, such as by radiation or a chemical mutagen. When we're talking about radiation, there are 2 types. The first is ionizing radiation, and these are radiation like X-rays and gamma rays. Radiation has a lot of energy. When this radiation hits a molecule or an atom, it will give the electron so much energy that that electron will leave the molecule or the atom. That will create an ion here because this will be a molecule with a positive charge. This molecule can then go on to react with other molecules, including DNA, and mutating the DNA. And that's the danger of ionizing radiation. Any question there? A non-ionizing radiation is radiation with less energy than ionizing radiation. And there's only one form of non-ionizing radiation that causes mutation because, literally speaking, light and microwaves and heat are all ionizing radiation, non-ionizing radiation. Only UV light causes mutations. Is that clear? How UV light causes mutations is when UV light hits DNA for 2 thymines, which are next to each other, right next to each other, the UV light will cause the 2 thymines to form a thymine dimer. Let me blow this up if I can. Where there will be 2 covalent bonds between the thymines. Why it's called the thymine dimer. There are 2 covalent bonds formed by the UV light between the thymines. This will interfere with the thymines. They will not pair correctly with their complementary base in the second strand of DNA. And then they obviously are paired to each other with a thymine dimer. That would create a bubble in the DNA. This bubble will cause problems for the DNA. This region of the DNA cannot be transcribed and it cannot be replicated with the thymine dimer in it. The thymine dimer has to be removed and then new bases put in, the correct basis in this case, before this region can be transcribed or replicated. And that is how UV light causes mutations. Now, if the DNA is trying to be replicated, the DNA polymerase will move along the DNA to here, will stop at the thymine dimer, and will not ever proceed further while there's a thymine dimer there. If the cell then goes through replication, it will have a mutation because this portion did not get replicated. Any question about that? I'm not going to talk about light repair. You will not be quizzed on it. Let's talk about chemical mutagens. We're only going to talk about one chemical mutagen. Nitrous acid is a mutagen. When it gets on DNA, it mutates the DNA. So here's the normal DNA. There's nitrous acid. Nitrous acid changes this A so that this A is not read by the cell as an A. In fact, the DNA polymerase will think that A is actually a G. So the DNA polymerase will read that changed or mutated A as a G and insert a C there. Then when this is replicated, that mutation will be fixed. You'll always have the G and the C match. Obviously, this can continue this way, although it's possible the cell can recognize this A is mutated and pull it out and correct it. The trouble is this A is the original DNA, and the cell, if it finds that this is not pairing correctly, usually will pull out this C and then read this and just put the C right back in again because it doesn't read that as a A, it reads that as a G. So this is hard to correct. Any question about any of this? Anyways, that's how nitrous acid works as a mutagen. It changes the nucleotide. So that nucleotide is still there, but it's changed, and the cell cannot recognize it as an A. It thinks that is a G. And so when the DNA polymerase comes and replicates this, it won't add a T there. It'll instead add a C. And that's the way nitrous acid works as a mutagen. The rate of spontaneous mutation is DNA polymerase makes a mistake about once in 10 to the 9th replicated bases. So in a billion bases that DNA polymerase replicates to make the new strand, one of those will be a mistake. Since about 1,000 bases is your average gene, at least in E. coli and most prokaryotes, that means the spontaneous mutation rate will be about 1 in 10 to the 6 replicated genes. Mutations increase this mutation rate by 10 to 1,000-fold extra. And that would increase the mutation rate from 1 in 10 to the 6 genes to 1 in 10 to the 5th genes down to 1 in 1,000 genes will be mutated in the presence of a mutagen. Any question about any of that? All right, we can test for the production of mutants by identifying the mutant experimentally. There are 2 ways to test. There's a positive or direct selection and a negative or indirect selection. A positive direct selection detects mutant cells because they grow or appear different. So if anyone were to watch this walking down the street, they'd say, yep, there goes a mutant. And that would be a positive direct selection. Any question about any of that? All right, a negative or indirect selection detects a mutant cell because they do not grow. I've actually selected for mutants in both ways. Let me explain a negative indirect selection in a little more detail. You can plate a colony, a plate of colonies. And what you do is you have your colonies dispersed, individually separated, and then you get a velvet stamp and you press it into the plate. That means that some of the cells will be transferred onto the stamp. So that one there will be that one there. This one here would be that one there. That one there would be this one there. You then take this velvet stamp and press it into 2 plates that have no growth on them. One plate contains the amino acid histidine. The other plate can lack the amino acid histidine. You then look at the colonies that grow up on both plates. And these colonies should be the same as the stamp here. So that would be X here, there, and then that one's there. That one's there. This one is there. And that would be the same as this plate here. You look for the presence of colonies on the media containing the amino acid histidine. And there we see a colony that is absent on the plate of media that is lacking the amino acid histidine. So these cells are mutated and they cannot grow on a plate. Come on, mouse. These cells are mutated and they cannot grow on a plate of media that lacks the amino acid histidine, but they can grow on a plate that has the amino acid histidine. And so these cells, where is that there? Probably this one here. These cells can grow, and they are mutant in that they do not grow if they are not supplied the amino acid histidine. That's negative selection. Looking for a mutant. Any questions about negative and positive selection for mutants? If not, let's talk about recombination. Recombination is an exchange of genes between 2 DNA molecules. It can be any exchange. It could be half the chromosome. It may only be a few nucleotides. Most likely it's going to be a gene. Crossing over is a specific case of recombination where the 2 chromosomes pair up, the blue chromosome A being the father's chromosome, having the lowercase alleles, and then chromosome B from the mother. So this could be the father's first chromosome, the mother's first chromosome. And it has the white alleles with the capital letters. So in crossing over, the 2 chromosomes pair up, And those are always homologous chromosomes, meaning the mother and father's chromosomes pair up. And then they cross over like this, and then they break right here, where now the father's chromosome, which is the lowercase here, the color's changed, is now lowercase e, lowercase f, lowercase g, and then oh, here it is. Sorry, lowercase e, lowercase f, lowercase g from the father's chromosome, and then capital H, capital I from the mother's chromosome. Sorry, the change in the colors kind of thrown me a little bit, but so crossing over is where the chromosome literally come together, cross over, and then break and recombine. Crossing over always involves recombination. Recombination is a little more general, and it does involve crossing over, but you can get recombination from another means, such as when a small region of one chromosome moves from that chromosome to the, let's say the mother's first chromosome. And it could be from the father's chromosome, a small region breaking off and then joining to the mother's chromosome, or it could be from another chromosome coming and then joining to the mother's chromosome number B. So that would be an example of recombination that is not involving crossing over. Most cases of recombination do involve crossing over, but not all cases. Any question about that? Recombination is important in that it brings different combinations of genes together that were not previously together. Like the capital G and the capital H are together, the lowercase g and the capital H are not. After crossing over or recombination, you can now have the lowercase g with the capital H. So recombination reshuffles the genes and makes new combinations of genes possible. Let's talk about genetic transfer. Cells can transfer DNA between generations of cells, and that's called vertical gene transfer. But cells can also transfer DNA between cells of the same generation. And this is an example of horizontal gene transfer, where DNA leaves the donor cell and then enters the recipient cell. There are three ways that horizontal gene transfer can happen: by transformation, conjugation, and transduction. In transformation, DNA leaves one cell and then is naked DNA in the environment, which can be taken up by another cell, which we'll call the recombinant cell or the recipient cell. And then part of the DNA that's taken up can be combined with the chromosome or exchanged with the chromosome. And then the cell can now express this new gene, the lowercase a gene, that was not from the DNA of this cell. It was from the DNA of another cell, which that DNA left and went in the environment. This is important in the case that lowercase a would be something like a gene for antibiotic resistance. This cell has now acquired the gene for antibiotic resistance. The frequency of a cell acquiring a new gene by transformation is about 1 in 10 to the 5th, at least for a bacteria. And the frequency of mutation causing that gene would be 1 in 10 to the 6th, meaning 1 out of 1,000,000 cells would be mutated, causing, let's say this is a gene for ampicillin resistance. And then the frequency of transformation is 10 times higher. So this cell can pick up the gene for ampicillin resistance by transformation, and that'll be 10 times higher than the cell getting it from mutation. Any questions about any of that? All right, we're not quite finished with this lesson. I've got, close to 10 different cells to go. I really wanted to finish transformation, but we are out of time. And so if there's no questions, I'm going to stop here. Any questions? All right, those of you who are in the 7 p.m. lab, I'll see you at 7 p.m.
Thank you.
All right.